Hi everyone, welcome back to my channel. After a long time I am meeting you all here. It's been already a month. Sorry for the delay. Today we'll be continuing the biotechnology wherever we left with. So in the last class we had studied about what is recombinant DNA. In this class we will be studying about how the recombinant DNA is going to combine with the host DNA. So in a chromosome there is a specific region which we call it as origin of replication. So you cannot insert this vector along with the foreign DNA in any region. You can insert it in a specific region called as origin of replication. Both in prokaryotes as well as in eukaryotes you will be seeing origin of replication. So origin of replication will be usually rich in adenine and thymine. Since they will be consisting only two hydrogen bonds, it's easy to break them and separate these strands so that you can insert a new strand. So if we have to insert a foreign DNA, then we have to cut the host DNA at specific locations. That cutting of these DNA will be done by enzymes called as restriction enzymes. So cut DNA plus vector, usually when you cut a foreign DNA and join it with the vector, there should be something which will seal them. That sealing property or the sealing job will be done by enzyme called as DNA ligases, and then this cut DNA along with vector, after they are sealed with the help of DNA ligases, they will be inserted into the host. The next topic that we are going to study is tools of recombinant DNA technology. What are the important tools that are required to create a recombinant DNA molecule and insert into the host? So we need restriction enzymes. So you guys know very well, to cut DNA at specific locations, we need these enzymes called restriction enzymes. Then we need cloning vectors. So after the alien DNA is cut and then it has to be inserted into cloning vectors. Then we need competent host. Once again, origin of replication in the competent host will be cut by restriction enzymes. And why this competent host is required? Host is an organism which is going to accept the foreign DNA and it is going to express that particular character in it. So this can be achieved with three of them. So these three are the tools of recombinant DNA technology. Now let us learn what is this restriction enzyme and where is it found and what does it do. So there is a small introduction here. W. Arbour in 1963 isolated restriction enzymes for the first time in bacteria. So you should remember this scientist name and his discovery in the year 1963. Later, W. Arbour along with two other scientists, Smith and Nathan, were awarded Nobel Prize for the discovery of restriction endonuclease. Endonuclease is one of the type of restriction enzyme. The discovery of this enzyme restriction endonuclease was one of the very important steps in recombinant DNA technology. This led to the successful invention of so many things. So these restriction enzymes, whichever helps in cutting the DNA at specific locations, belong to a class of enzymes called as nucleases. There are two types of nucleases we have. One is restriction exonucleases and another one is restriction endonucleases. Exonucleases, exo means outside, it starts to cut the DNA from the ends. Whereas endonucleases will recognize specific points inside the DNA and it will start cutting the DNA at those locations. Exo means outside, endo means inside. Hind2 was first restriction endonuclease isolated from the bacteria Haemophilus influenzae RD. RD refers to the strain of the bacteria. It cuts DNA at specific region by recognizing specific sequence of 6 base pairs. So it will find out a specific region where it can start to cut the DNA, and that specific region will consist of 6 base pairs, and hence it is called as recognition sequence for Hind2. So it is the sequence that Hind2 is going to recognize and start to cut, hence we call it as recognition sequence for Hind2. Let us now learn how to name these restriction enzymes. So restriction enzyme's name will be derived from the bacteria through which it was obtained or through which it was isolated. So first letter of the genus name. So whatever bacteria we have, its first genus name whatever is there, the first letter from that genus name should be taken out. Second two letters of species name. So in species name, what first two letters we have, that we have to take it out. First letter of the strain. Strain is the type of the bacteria that is present. So in a bacteria with genus name and species name, several strains of bacteria will be present, which will have some minute differences. Like in case of Streptococcus pneumonia, we have two types of strains, rough strain and smooth strain, which we studied in molecular basis of inheritance, isn't it? So those are the two minute differences, the type of colonies which will be represented there. So likewise, we in other bacteria also we have strains which vary at minute differences, so that we call it as strain. So first letter of that strain will be obtained, and then later Roman numbers that indicate the order in which enzymes are isolated from that strain. Maybe that enzyme would have obtained first from that strain, or third from that strain, or second from that strain. It depends, right? So that order we have to mention in the end. For example, if you see Escherichia coli, RY13 is the strain of Escherichia coli that we have taken here. So from Escherichia we are extracting E, that is first letter of the genus name. Then from coli we are extracting first two letters, that forms CO. Then this strain's first letter is R. So we are taking R, and the Roman letter of which the enzyme was isolated, whether it was isolated first, second or third. It was the first enzyme that was isolated from this specific strain of bacteria. So we mention it as 1. So that is why we call it as ECOR1. It is the enzyme obtained from Escherichia coli RY13. For your better understanding, I will be telling you guys some other examples too. So Bacillus amyloliquefaciens H. How will you write this? BAM, okay, B, A and M, which is written in italics. First three letters should always be in italics. Then its strain H you will be writing. Then the order through which it was obtained. So BAMH1. Same way for Haemophilus influenzae RD, you can write it as HIND2 or HIND3. Here instead of R we are considering D, that is the only difference, so that the pronunciation will be easy. Then Proteus vulgaris PVU, and obtained in the second position, so PVU2. And Providencia stuarti PST1, Thermus aquaticus TAQ1, Streptomyces albus G SAL1, Escherichia coli RY13 ECOR1, Escherichia coli R245 ECOR2, Escherichia coli J62PL G74 ECOR5. If you see, these genus and species name are same, but there will be slight differences in them, the minute differences, which we mention it as strain. And RY13 was obtained first, hence 1, and R245 was obtained second, hence 2, and J62PL G74, from this it was obtained at fifth position, hence we write it as ECOR5. This is how the restriction enzymes are named. Usually the restriction enzymes are of three types. Type 1 and 2, we can't use it in rDNA technology, so I don't want to deal with that in detail and scratch our heads. So it's better we get into the one which we use in rDNA technology. So here type 2, it requires magnesium for its activation so that it can do its restrictions job. Apart from that, it is used in rDNA technology. So they might ask this for MCQ. Apart from this, this is very important. ECOR1 and HIND3 produces sticky ends. So when they cut the sequences at particular region, the ends will be sticky. So when ends will be sticky, if at all there are any complementary bases, they will stick on to each other and it will produce a more efficient rDNA. Okay. whereas ECOR2 and Hind2 produce blunt ends. Blunt ends means it will not be sticky, and here you might not have complementary bases, right? In that case we can use ECOR2 and Hind2. And if at all they ask which is the most efficient one for rDNA technology, it is going to be ECOR1 and Hind3, as they produce stick ends. Sticky ends are more efficient than the blunt ends. In case you don't have the complementary bases there, then you can use the ECOR2 and Hind2 for inserting your gene of interest and or your complementary bases, so that it can attach with each other. And ligation is much required in case of ECOR2 and Hind2, whereas ligation or joining of the two ends will be very easy when sticky ends are produced.